Hello dear students, welcome to the rapid revision series of Science and Tech. I know there has been a little bit of delay. I was not keeping well with health. That is why there was a delay. Nevertheless, what I've planned for this session is, because we are very close to the current prelims which is there, we will be doing a very priority based revision of Science and Tech. As in, like, not all areas in Science and Tech deserve to be, revised again and again and again. There are certain areas which have to be in your memory, and there are certain current events that have to be in your memory right before the exam. Okay, so this is what we are going to do. So it's going to be a very quick, but the important points will be covered. So what I'm going to do is, along with this, after this session, the PDF that gets shared with you guys in the official group and everything, that will have more slides and more content for those who want to just keep reading and maybe go through it. The lesser important topics that, if you just want to go through it, or some people would have the tension that, okay, have I covered all the important. So some people who have the tension that, maybe have they covered all the, all the current affairs that is there for Science and Tech, that kind of a tension if you have, and, that's holding you back with respect to the subject, that lesser important topics will be there in the PDF. But those who want a quick revision of the most important topics, I would advise just watch the video, put it in 2x speed. Already I speak very fast, so you can just get it over with in one sitting itself. When, for example, if the video gets uploaded today, you can just get it over with today itself. So that's how I have planned this session. So understand, those who want more clarity on it, you have to take the idea that, like, for example, let's go about with the topic, and you will see that maybe some areas we will not go in depth and into the basics of it. That ideally need not be covered at this point, because it's very close to the exam. So this is just like I would say this is like a crash course for the most important things that you need for Science and Tech. Okay, so without much delay, let's go into the first area, and obviously the most important area when it comes to the kind of area that gives a lot of nightmares to a lot of students, which is the area of biotechnology and related biology associated with it. So we start off this topic with, normally when it comes to, let's have a quick idea about this. See, when it comes to biotechnology, one of the first topics that are important are your differences between your animal and plant cell. And here, understand that fundamentally there has also been a question asked with relation to it, but what you have to take from here is the core differences between an animal cell and a plant cell. So when you look broadly, there are these two types of cells, and one important pointer, let's start from the size. Your animal cell is considerably smaller in size than your plant cell, which means that your animal cell would be somewhere into 10 to 30 micrometers, whereas your plant cell can go from 10 to almost 100 micrometers. So when you look at the size difference between animal and plant cell, your plant cells are much more larger in size. That's the first thing. Second important difference comes in the form of what kind of materials are there in the cell. As you know, the cell, when it comes to an animal cell, is quite irregularly shaped, and the plant cell has a very fixed, rigid shape. The shape is obviously a difference, but added to the shape, the reason why there is this shape difference is because the plant cell has an important organelle called as your cell wall. When I'm saying organelle, understand that these are smaller subunits of your cell. What constitutes a cell is called as an organelle. Those who are from non-science background, don't think it's some Greek word, okay? So this plant cell has a cell wall, which makes it quite rigid in shape, whereas the animal cell does not have a cell wall. The next important difference is the plant cell has something called a very large unit in it called as a vacuole. Okay, so this vacuole is primarily used to store a lot of lipids, oils, etc., whatever is produced in the cell. Now, I want you to remember, The animal cell also has vacuoles, but they are very small in size. So it's not the presence or absence of vacuole, it's that both of them have vacuoles, but in the case of the plant cell, the vacuole is quite large, and in the case of the animal cell, they are smaller in size. Okay, the next important organelle that the animal cell has, but the plant cell does not have, is something called as a centriole. Okay, so the centriole is present in the animal cell. The centriole is an organelle that helps in cell division. Okay, so understand here what happens is, in the case of the centriole, it's present in animal cell, but in the case of plant cell, the cell division has a little bit more additional processes. So what happens is, here there is no centriole. Okay, so centriole, centrosome, when you hear any of these words, associate it with the animal cell and not with the plant cell. Okay, so these are the primary differences which are there when you look at it. And obviously the plant cell has a certain plastid called as your chloroplast. Obviously this chloroplast is not present in your animal cell. Why? Because you do not need, the plant cell has this chloroplast which will help it give the chlorophyll that will help it to produce its own food. Animals do not have that function, and the reason why they do not have this function is because at the cell level they do not have this plastid chloroplast which will produce your chlorophyll. Okay, so primarily these are the differences, but I want you to remember this is a topic that will get, like for example, each time, now the next time I'm going to talk about maybe stem cells or something like that, there will be a difference with respect to that. So keep this point in mind. I wanted to keep a table differences, plant and animal cell, add the points that I just told you, but keep adding the points to it as we go further in the session. Now coming to another type of cell which is there, which is your prokaryotic and eukaryotic cell. Understand, when I'm saying another type of cell, it's not outside the world or anything. It's in here itself. But prokaryotic cells or prokaryotic organisms are those organisms which do not have a very defined nuclear region. What is the meaning of this? So here when I told you about the plant cell and animal cell, I told you only the differentiating points. Obviously, there are certain organelles which are common to both. And one very important such organelle that is common to both animal and plant cell is the center head of this cell called as your nucleus. Got it? In both plant and animal cell has it. Inside the nucleus, you have the majority of the cell's genetic material or our genetic material as it is, which can be DNA, RNA, whatever it is, it's all contained inside the nucleus. Got it? Now, there can be certain organisms where, can you see here when I'm saying nucleus, it's clearly bounded. I just went about and drew a circle and then I marked it as nucleus. Why am I able to do that? I'm able to do that because there is a clear region that is demarcated by something called as a nuclear membrane. Okay, so there's a clear region called as nuclear membrane. Inside the nuclear membrane, everyone, nuclear membrane is like, okay, fine, come on, all the DNA, all the genetic material, everyone come inside, and the nuclear membrane just covers it like this. But in some organisms, this nuclear membrane is not there. So when the nuclear membrane is not there, what is going to happen? Everybody will like, okay, fine, genetic material will be there, the other things will be there. So everyone will just float around in the cell. So this exactly is what happens in the case of a prokaryote. In the case of a prokaryote, there is no defined nuclear region or any kind of membrane-bound cell organelles. All of those are absent. So you can understand, prokaryotes are also those organisms which are much less evolved. When I'm saying less evolved, don't just conclude in your head like, okay, yeah, bacteria, algae, fungi, all of them are prokaryotes. Not necessarily. Even amongst bacteria and algae, there are organisms which are eukaryotic, which means they have a clear nuclear membrane. They have a clear nucleus. So what is your conclusion over here? When someone tells you all microorganisms are prokaryotes, don't believe that. Why? Because microorganisms can be eukaryotes also. When someone says all prokaryotes are unicellular, need not be. What is unicellular? That they just have one cell. Need not be. You can have prokaryotes which are multicellular. You can have eukaryotes which are unicellular. So don't believe in such generalizations. Don't go for such generalizations, prokaryotes and eukaryotes. Just the differentiation is based on the absence and presence of nuclear membrane. I keep pointing out this because I do expect questions from this area, because it's a very basic thing, right? Which non-science students might always leave out. Okay, so I hope this part is clear. Now we go into an important topic even for this year, which is your stem cells. So now, where I have to discuss with you stem cells, understand the basic of it lies from the fact that in every human organism, that human organism, plant organism, whatever it is, you will find two types of cells. You will have something called as your somatic cells, which are otherwise your growth cells, and then you will also have something called as your germ cells, which are your reproductive cells. A quick revision into this, understand your growth cells are what has created you like this, like you have two arms, you're constantly increasing in height. All of these things are because of your growth cells. They constantly replicate and they create many multitudes of cells. What is important for you to understand is, in a growth cell, we all keep saying a human has 23 pairs of chromosomes, right? 23 pairs of chromosomes, which means in total we have 46 chromosomes. I want you to understand that in the case of a somatic cell, they will have, let's put it like this. This is where you have to understand, in the case of a somatic cell that is there in your body, which is present all throughout in your body, all of them will have 23 pairs of chromosome. 23 pairs. The reason I am specifying this is because in a germ cell, that is a reproductive cell, the reproductive cell is different in organisms which are called as male and organisms which are called as female. What happens is the reproductive cell is also different. In the case of females, the reproductive cell is something called as an ovum, and in the case of males, the reproductive cell or the germ cell is something called as a sperm. Now you do understand that ideally, if a human fetus has to be created, you need germ cells from both the human female and human male. Which means what? In the case of now, in my body, if there is a germ cell, how many chromosomes will be there in it? It will not have 23 pairs. It will just have 23 chromosomes. Why? Because from my end, there will be a 23 chromosome. From another person, there will be a 23 chromosome. Together, it will make a 23 pair chromosome in the human fetus. Are you getting the point? So here, understand, when they ask you the primary difference between a somatic cell and a germ cell, and they start talking about the chromosomes and everything, understand, in a growth cell, you have 23 pairs. In a normal germ cell, you have 23. That's it. No pair. Okay, 23 chromosomes. Now, let's assume, okay, let's begin the process of creation of a human fetus. And at this point is where I want you to tell you. So I'm going to clear this slide at this point. Okay. Now let's assume that a human female and a human male have contributed their germ cells and they've created a single cell. They've fused together, they have fertilized, and what has been created? A cell which has now 23 pairs of chromosome. And this is something that we call as zygote. Now imagine we all were at one point a zygote, right? We were all a one-celled creature at some point. From where? From that one cell, it became two, and the two became, each of them became again two. And in this manner, finally, a human fetus gets created. When I'm saying human fetus, a human fetus is not like a shapeless ball of cells. A human fetus has eyes and mouth and head and brain inside and heart and hands and everything. So now is where you have to understand there's a very important process that happens for a zygote to become a completely formed fetus. This process we normally call, the development of the zygote. The development of the zygote is possible because of a process called as differentiation. Differentiation. Okay. Now I know it's quite unclear right now what is the differentiation. And here is where I want to introduce you to something that happens in the human body. Ideally, now what has happened is you have a zygote. Okay. Now the zygote, as I told you, will start replicating the cell. It's a cell, right? So it will become 2 and then 4 and then 8 and 16 and 32. In this particular stage, when there are maybe 16 cells or 32 cells, when you look at those cells, these cells are not like the normal growth cells that are there in our body. These cells which ideally at this point we are going to call it as stem cells, they have the ability to become any organ possible. Okay. I always think of stem cells at that stage as some kind of, they have some kind of a superpower in them. Why? Because you take that stem cell, the stem cell can turn into a heart. That stem cell can that same stem cell can turn into a hand. That same stem cell can turn into a brain. So understand, the cells which are at that 16 to 32 cell stage, they are like, I can be anything in the world. I can be any organ in the world. And this process by which they can be anything, any organ in a human body is called as differentiation. So if I had to ask you, those cells at the embryo stage, at the 60 to 32 cell stage, they have the highest possible levels of differentiation, because they're like, brain, okay, fine, done. Heart, okay, done. Okay. They can be anything that they want. So this process of your unspecialized stem cells becoming specialized cells is called as differentiation, which means what? Unspecialized means what? They do not have a particular idea like, I want to be brain or I'll do the function of brain, nothing like that. Now, because they can be anything, what happens is they undergo differentiation, and once they undergo differentiation, they turn into a specific organ. It is in that manner where from the zygote stage, once the human fetus is completely formed, you have all the organs in it. So it's at this stage that they decide like, okay, fine. Now, when you ask me, they decide, understand, each cell at the 16 to 32 stage has the ability to differentiate, because at that stage, inside the cell, it has a specific genetic code. Now, don't think too much into this. Just keep in your mind, it has a specific genetic code that has the ability, or that prompts the cell, like, be anything you want. And we all wish we had that. Now, what you have to understand is, as we grow further and further old, we do not have a lot of cells in our, when I'm saying old, the fetus comes out, right? Then it becomes 1 year, 2 year, 3 year, 32 years, 40 years. As we go further, what happens is, this stem cells are not going to be there in our body in very huge numbers. The highest number of stem cells you will find at the embryonic stage. 16 to 32 cells inside, when it's there inside the mother's womb, inside its fetus stage. At that point is the highest number of stem cells and also the highest differentiation potential when it comes to an animal cell. Here is where I want you to specify this. We are now, since I'm talking about humans and everything, you have to get the idea that we are talking about stem cells in animal cells. Okay, so ideally, if I had to broadly look at it, there are two types of stem cells, which is the one that is there at the embryo stage. They are full of power. They have the ability to differentiate into whatever possible. They also, some remains of it. Okay, so at the 16 to 32 cell stage, it will develop in everything. At the end, once the fetus comes out, even in the umbilical cord, that connects the mother with the fetus, there also you will have some stem cells. Okay. Then after which, once the fetus grows, then you have stem cells which are called as non-embryonic stem cells or adult stem cells. Where do you find it? You can find it in the bone marrow, you can find it in the peripheral blood tissue, etc. Which means right now in my body, in the bone marrow of mine, there is stem cell. But what is its problem? As I told you, with age and with the processes of living and everything, what happens is the differentiation capability of your cells or your stem cells reduces. Which means if I had to compare the differentiation potential is primarily what? The ability to be anything. Just keep that in mind. So if I look at the embryonic cell, it's the highest at the embryonic stage, the ability to become whatever organ. But if I take my bone marrow stem cell and be like, bone marrow stem cell, let's try to make it into a brain cell. Bone marrow stem cell is like, I'm done. I've become old. I cannot do that much of activity. Try making me into another blood cell or something. Which means what? Here, the non-embryonic stem cells, it will have lesser differentiation ability. Okay. So even in animal cell, there are two different types, embryonic and non-embryonic. And this is where there is another difference between animal cell and plant cell. So as you can see in the animal cell, differentiation capability reduces with age. Okay. But in the case of plant cell, it's not like that. In the case of plant cell, you at any given point, if you take a cell, it has somewhat equal differentiation capability. That means if in the case of the plant cell, maybe at its embryonic stage and its grown stage and its old age stage, if I'm taking a cell, that cell will have equal differentiation capability in comparison to an animal cell, where the differentiation capability reduces with age. So keep this point in mind when it comes to your stem cell ability. Here in the case of plant cells, the stem cells in plants also have stem cells. In the case of stem cells of plants, the differentiation capability is equal. In the case of animal stem cells, the differentiation capability reduces. Now, the reason why I went so much and talked about your stem cells and everything is because we have current affairs associated with this. Okay, so until now, I also want you to keep in mind, we were just talking about natural stem cells, naturally in the embryo, naturally in the bone marrow, etc. The next topic that I want you to get to understand is, so when it comes to the natural process, whenever we are there are some features associated with it. One of the features being that you can preserve your stem cells that you're taking. I told you in the umbilical cord there can be stem cells. You can take it, you can preserve it in liquid nitrogen, in cord blood banks. Technically, there is no expiry date for stem cells because it's an organ and you're preserving it in certain conditions. It will become, or it will remain, or it will have its vitality, as in it will remain and it will have its life as long as that preservation thing is kept. But scientifically, when you look at it and when you look at how frequently you have to use it and everything, scientifically evidence suggests that they can be stored for about 20 years. So as of now, we have stem cells that have been stored for almost 20 years and have remained without any damage. Ideally, why do we have to do any of this? Why are we so bothered about stem cells? We're so bothered about stem cells because when you collect these stem cells from the umbilical cord and you keep it, at that point understand why in the umbilical cord stage, the stem cells do have the differentiation ability. It might have reduced. It might not be as differentiating as your embryo stage, but still it's not as pathetic as your bone marrow stage. So once in that umbilical cord stage, when you are taking and preserving the stem cells, in the future, if that a person develops any kind of disorders, any kind of blood related disorders or genetic disorders like thalassemia, sickle cell anemia, leukemia, any of these disorders, you can, where you need to replace the body's cells, what you can do? Go to the umbilical cell of that person which has been stored, take the stem cells, do the enough research in the lab, not research, propagation in the lab, put it back into the person's body. It can be a blood cell, it can be a nerve cell, whatever for which the person's requirement is. Got it? Now, until now, we were all talking about natural stem cells, which is there in the umbilical cord, embryonic stage, etc. Now, I told you, I clearly mentioned to you, even in your bone marrow, there is stem cell. Now, assume a person has, they have not stored the umbilical cord of the person. The person is met with a very life-threatening blood disorder like leukemia or something. Now, the only thing that's left in that person's body that we can actually use and, help that person is the bone marrow stem cell. But I already mentioned to you, the bone marrow stem cell is not has a very high differentiating capability. So here is where you understand you have another process called as induced pluripotency. Induced pluripotency. Now here is where you have to understand pluripotency means the potential of a cell to become multiple organs. Okay, so pluripotency will be highest where? In the embryo stage. When it comes to the human, pluripotency is very low when it comes to the bone marrow stage. So here what am I going to do is, here I can take the stem cell from the adult stage and I can change that stem cell or I can incorporate something into it. Now, I told you very clearly, an embryo is behaving in that stage or in that 16 to 32 cell stage. Why is it behaving like I can be anything? It's behaving because it has a certain genetic code in it, right? As we go further and further, that genetic code is not replicated often. That is the reason why in the bone marrow, we do not find that high ability of I can be anything. So what happens is, I look at, now you have to understand this. This is one doubt I've gotten in a lot of my biotech classes, but I want you to clearly understand there are methods right now in the world whereby which I need not necessarily take out an embryo or anything. I can scan the embryo's genetic code I can see what is the genetic code of an embryo. So obviously research has happened in all that front, and I know exactly now if an embryonic cell is behaving like in a very pluripotent manner, it's because it has some genetic code in it, and I also know what that genetic code is. Okay, so think of it like a secret thing where at the genetic code maybe there is a code AGC. Now I know this AGC is what is making this cell behave in this manner. So now if I just have that AGC genetic code in my bone marrow stem cell, that will also behave like that one, right? That will also, this stem cell will also start behaving like this embryonic thing. Why? Because you have incorporated that one thing that makes it different. So now this process by which you induce pluripotency. Induce means what? You are artificially giving it pluripotency. You can, using this process, you can take your human stem cells which are in the adult stage and turn it into a very highly pluripotent cell. So what is the implication of it? The implication of it is that you don't necessarily have to go and take embryonic stem cells anymore. I can take a human adult stem cell and create it, or you can induce pluripotency in it. Once pluripotency is induced in it, is there any difference between that and an embryonic cell? No. Which means what? Using that induced pluripotency, I can do the same activity. If someone needs some cure for sickle cell anemia or thalassemia, or they need extra cells in their human adult body, I can do it. So, induced pluripotency is the artificial method by which stem cells or your adult stem cells can be turned into pluripotent cells. Okay, and if all this is clear, this is the base that you need to understand what is the current affair in this field. And trust me, I've gone into such lengths to explain this because of the current affair. One of the first current affairs that have happened in this area is very recently, the stem cell therapy has shown very good results in HIV patients. I hope you have noticed in that list of diseases which is I have written sickle cell anemia, thalassemia, leukemia and everything, but I have not written HIV because until now it was not seen that we could provide a cure for HIV using this. But that has happened. So they could just very casually give you a question, stem cell therapy can be used to cure HIV. And I want you to understand, yes, it can be used to cure HIV. So what is happening in this process? Here what is happening is, again, here and this will, this was done in a patient called, the patient's name is Geneva patient. They do not leave out the patient's name for obvious privacy reasons. But understand, I want you to keep, I'll go into the details of it. See what happens when a person has HIV. In the case of HIV patients, the body's own immune system starts working against it. Why? Because the body's own immune system loses its ability to identify which is a foreign cell, which is its own cell. So what happens is, see, right now we all are healthy because if something foreign enters our body, our immune system is like, yeah, fine, go, hit it, kill it, okay? But what if the immune system is turned into such a creature where, or such a system where anything external also it will fight? And if a new cell is created in my body, see, constantly in my body, there is cell replication happening. My immune system is like, kill it, no need. Imagine that. So what is going to happen? Your immune system starts functioning against you, which means in your body, constantly cells are getting damaged. So using your stem cell therapy, what are you going to do? there's a lot of cells getting damaged in my body. Instead of those cells, using stem cell therapy, I pump my own new cells into my body using stem cell therapy. But I hope you do understand, it's not going to be something like a routine treatment for HIV. Why? Because it is going to take several rounds of such pumping back my cells into my body. So as of now, it is not developed into a routine treatment, but there is possibility to understand that using stem cell therapy, you can, and hear this word very clearly, you can suppress HIV infection. So if they ask you, HIV can be cured completely, that statement is wrong. HIV can be cured? Yes. Okay, so understand, be very careful. They can go into the technicalities in this manner, so keep that in mind. So this is the first current affair. The next current affair is, this is what I was telling you. Look at the name of this, HERVH gene. What exactly is this? I just told you that the human embryonic stem cell or the human embryonic cell is behaving like, okay, I can be anything, I can be pluripotent, I can be all of these things because it has a genetic code. Very recently, the scientists have gone and seen that this is the genetic code. There is a specific genetic code where they have named it HERVH gene. This HERVH gene, wherever it's present, what happens is, that cell will behave in a pluripotent manner. So now you know, if I have to induce pluripotency, what can I do? I just have to recreate that HERVH gene into the adult stem cell. Understand? So I also understand most of the inner mass cell means what? The ones that later on become body organs. They have your HERVH gene. So next time someone asks you, why is the human embryo behaving in an extremely high pluripotent manner? Understand, it is having the presence of your HERVH gene. These were both important discoveries with respect to stem cell therapy this year. This is the reason why I chose to discuss this. I hope this entire topic is clear to you. If it's clear, just mark in the comments yes. And I've been noticing your comments also. Somebody has asked like how to get in touch and everything. So you can do one thing, just email whatever is your doubts to the IAS Baba team. They will obviously convey it to me. And I can, through the organization, I can get in touch with you. Otherwise, if I give out my personal number, I don't think that will be a very wise idea. That is why, okay? So now coming into another area that we have to discuss, it is, even now we have not got into biotechnology. We've just started off with stem cells, but it's an important topic. So I hope that's clear. Then we go into something inside your nucleus, and this is where all the activity of biotechnology is happening. And this is where you have to understand, as I told you, there is a nucleus. Inside the nucleus, you have a lot of genetic material. And what exactly is the genetic material that we are talking about? We have, or most of the organisms that we know, have a genetic material inside the nucleus called as your DNA. Now, few basics to keep note over here. Do all organisms have DNA as its genetic material? No. There are organisms which have RNA as its genetic material. Okay. Now, the difference between DNA and DNA and RNA, I hope you know it. DNA is double-stranded, RNA is single-stranded like this. DNA has something called as different nuclear base pairs which are there. That is what makes the structure in this very nice, rigid structure which is there. So there is cytosine, guanine, adenine, and thymine. In the case of RNA, there is no proper structuring with, that two base pairs are not held together or anything. They just singularly put like this. So here you will see cytosine, guanine, adenine, and instead of thymine, you have another thing called as uracil. I want you to also understand that these base pairs, they are called base pairs for a reason. The reason being that they're always as a pair. They are best friends forever. So cytosine and guanine, you will always, so if there is a cytosine, there will be a guanine paired with it. If there's a guanine, there's a cytosine paired with it. So it will not be like, okay, one day cytosine will be like, let me go and see what's friendship like adenine with. Nothing like that. They will not pair together. Okay. So these are the base pairs. These are the base pairs. Got it? Now coming back to my original question over here. See, so each organism has its own type of genetic material. Okay. And this is the first important thing I want you to get clarified with. So there's this common idea that all your bacteria, virus and all has RNA. Nothing like that. Okay. In viruses also, there are viruses which have DNA as its genetic material and RNA. So it's nothing, you cannot universally say that, okay, DNA means it will only be for them. Nothing like that. But a very important point that you have to keep in mind is, we'll come to it later also, but just remember in your mind, if there is an organism with RNA as its genetic material, and there's another organism with DNA as its genetic material, at no point will they both have a progeny. They both will not have any kind of fusion between them. They both cannot, I cannot be like, okay, this RNA is only one strand. No, I will take one strand from here and put nothing between them. When the genetic material, that genetic material is the most fundamental feature of any organism. If the genetic materials are different, then you cannot have fusion, you cannot have cutting and erasing, cutting and putting together. None of that is possible. What I mean to say is, you cannot cut a strand of RNA and put it into DNA, and you cannot cut a strand of DNA and put it into an RNA. Why? Primarily because they are so different. DNA is double strand, RNA is single strand. DNA has thymine, RNA has uracil. So any question which says an organism which has DNA as its genetic material and a virus which has RNA as its genetic material, can they be made into some organism? No. No. Got it? So keep that base point in mind. Now I know scientists out there and biotech experts amongst you like, no ma'am, there is possibility to do. I'm talking from UPSC point of view. Just keep this in mind. Okay? Please don't overcomplicate things for UPSC. The thing being that they just take these things in a very general tone. Okay? Now coming to these both parts, I also told you one pointer over here that the DNA or the RNA, whatever is the genetic material, is stored in the nucleus. And this is where our next current affair is going to come as important. Understand, first of all, the first thought that you have to take away from your mind is, not all the genetic material is stored in your nucleus. Okay, maybe 99% of the genetic material of an organism is stored in the nucleus. When it comes to animals, a small part of it is stored in mitochondria. And this is where our other current affair, which is important for this year, 3-parent baby and everything is important from that point of view. So from an animal's perspective, so if you have a cell, nucleus has 99% of your genetic material, mitochondria has a little bit part of your genetic material. Why did I specify animals? Why? Because in the case of plants, this same extra storage is done with chloroplasts. That means in the case of plants, 99% in the nucleus and small part in your chloroplasts. Okay, so until now, so this is what we have to understand. Cell, the genetic material, be it DNA or RNA, whatever it is, it's not just in your nucleus. A little bit part of it is in your mitochondria or your chloroplasts. Okay, so I hope that pointer is clear. Then comes the current affair that is there. This year, there has been an extensive research in something called as cell-free DNA or cfDNA. What exactly is cell-free DNA? See, now obviously in our body, as I told you, there is constantly cells are dying, new cells are created, etc, etc. Okay, so what happens is sometimes when the cells break down and everything, fragments of DNA, pieces, small pieces of DNA which is there in the nucleus, they can be present in the bloodstream of our body. Is that DNA inside the cell? No, it's not. It's cell-free. Why? Because the cell has broken down, the nucleus has broken down. When I'm saying broken down, expired, it's broken, okay? And it's just floating. It might be floating in our blood, when we, in the urine, any kind of secretion that we have from our body. All of those, you can expel or you can have those DNA fragments. So this DNA or the fragments of DNA released from the cells and are present outside the cell in the body fluids. Body fluids means what? Blood, urine, semen, etc. In all of these things, in the body fluids secreted out of our body, there you can have presence of DNA. Got it. This is also the reason why when there are traces of this out at a crime scene or anywhere, they're able to track what the DNA is it from these elements. Why? Because they'll have fragments of DNA in it. Okay. This area, see, this is all, it's not something new. We always knew cell-free DNA was there. We were aware of it since 1948. But very recently, what has happened is there's a lot of new applications coming on it. And when you hear applications, you know very well. UPSC will want to ask a question about it. So what are the different applications that are known? See, one of the most important applications of your cell-free DNA is, first of all, understand the possibilities of it. Normally, if I have to do any research on DNA, I have to go to the cell. I have to go to the nucleus. I have to go inside the nucleus, find out the DNA fragment, which is any kind of research which is there. But here, without doing any of it, the DNA is just floating around in the blood. I just have to figure out the DNA and then look, if I have to do any research, if I have to collect anything, I just have to figure out where they are. So understand, one of the most important applications of it come in the form of screening of fetuses. In a lot of times, what happens is, whenever you have to test if the newborn baby or if the coming newborn baby has any kind of genetic defects or anything, what generally happens is, they try to collect the fluid which is there in the, mother's, or they take out certain parts in the mother's uterus, which can actually be quite risky, okay? But they do it so that, you can screen for if there's a history of genetic deformities in the family or anything. They want to make sure. So this is where they do this process. But it's invasive. Invasive means what? You are going inside and collecting something. It's risky, etc. Now, you don't have to do that. Why? Because you can look at whatever is in the bloodstream when the urine the mother passes or any of these things, because that will have even the compounds of the fetus. So I just have to look at that DNA content or that genetic material, and I can look for, or I can do non-invasive prenatal testing. Non-invasive prenatal testing is primarily that I don't have to go and take fluids from the mother, whatever is there in the form of cell-free DNA, I can look at it and I can get an assessment of the genetic abnormalities. So through this, even in the form of early detection, diagnosis and treatment of cancers, understanding why a body is rejecting a transplanted organ, as a biomarker for neurological disorders like Alzheimer's, neurotonal tumors, stroke, traumatic brain injury. In all of these cases, I just have to look at what kind of genetic material is there in my bloodstream instead of waiting. For example, in the case of cancers and all, a lot of people detect cancer only when there's an actual tumor or anything that is present in some part of their body. But actually, you can, if you're looking at the cell-free DNA in your body, you will always understand if any kind of errors have happened in your DNA. Because you have a lot of breakage, you have a lot of fragments in the bloodstream, you can detect it even prior to you forming a tumor or anything. So that is why early detection, diagnosis and treatment of cancers. This is an important current affair. Okay, because there's a lot of focus given on these kind of new applications. So I hope that is also clear. By now, I hope you have understood the next. So by now, you've also got an idea about how this session is going to go. So I am talking about, I mean, I could very well take all my time and discuss about all of the important, what you say, not important, all of the basics of biotech and everything, DNA, fingerprinting, this and everything, but that's not necessary right now. This is very close to the exam. So we are just dealing with the current affair that is most important and the background, because every single time UPSC, when it comes to biotech or science and tech questions, never asks just like a current affair question. They always related to the basics. This is the reason why I'm dealing it this way. So I hope you are fine with it till now. If it's not fine for you or the way the session is going on till now, I would advise you, please don't go further and waste your time or anything, because this is meant for this kind of a revision. Okay. So now coming to the next part that we have to discuss, here is where I want you to discuss one important factor that is there. So I told you we all have DNA in our nucleus and our mitochondria, etc. We'll talk about the 3-parent baby later, but right now, understand, so we all have DNA in our body. Now what happens is, and this is the basic of all kinds of biotechnology, see, the DNA in our body, just by being over there, it's not doing anything for us. Why? Because DNA means, as I showed you that double strand, it's actually several kilometers of genetic material that is out there. Okay. Now, not all of that genetic material is actually useful. The only certain parts in this genetic material, maybe for example, certain parts, when I'm saying certain areas, certain codes, which are actually functional. Rest all of it is like, have you seen these old, I mean, I'm pretty sure you guys would not have seen when you had your typewriters and everything, to just get the typewriter in the loop, they would just type random gibberish like X, Y, Z, 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 so a lot of things. And then once it's on track, then you will have a word that is there typed out clearly. Now, why are they doing it? The thing is that here you, first of all, when you are having this just simple codes over here and here, or whatever is useful, only the entire focus of the cell is to get that into proper manner. Okay, which means what? In our human cell also, several kilometers of DNA will be present, but only some parts of the DNA are actually useful to us. Those useful functional parts of the DNA are called as genes. Understand? So also understand one more thing. This is also the reason why scientists and everyone tells you 99% of human DNA is similar. Between you and me, the human DNA is similar for almost 0.4 some percentage which is there. The only difference is the rest part, and the rest part is the most important part. Why? Because the rest part is what makes you and me and another person all different from each other. And that part is your genes. Just because, so I hope that's clear. A lot of students use DNA and genes very interchangeably, and that's all right also, if you're looking at it from a very non-science point of view. But understand, the gene is the functional part of your DNA. Now that doesn't end over here. When I'm saying it's functional, how does it become functional? And this is where you have to understand. So let's have a look at this. This is what your DNA looks like. Okay. Now, as I told you, there are certain parts of the DNA that are useful for us. When I'm saying useful, it means that when in our body that part is there, and that in that DNA that part is there. Now maybe it's happening right now also in my body, where what happens is DNA, which is normally coiled in this manner, and here there is one useful part that is there. When I'm saying useful, it has to become functional. So what happens is, one point, the DNA is like, okay, let's start making it useful. So what happens is, this part, it will start uncoiling. Uncoiling means what? It will become like this, so that that part can be clearly seen. Once it's uncoiled, or that part becomes clear, then let's assume here there is something. So adenine is there, thymine, guanine is there, cytosine is there. So guanine. These pairs are there, and this is the useful part. Once they have uncoiled, what next happens is, once they are uncoiling itself, the body gets the message like, okay, yeah, time for the functions. And at that point, what happens is, a RNA gets created in your body. Okay? What RNA? This RNA is called as messenger RNA or mRNA. So now what has happened over here? There was a DNA. The DNA got uncoiled. Now here, once it got uncoiled, the body starts creating an RNA over here. It's not RNA got from export or import. The body created an RNA which is complementary to this. Complementary to that first strand. So what is this? A is there. Ideally, what should come? Thymine should come. But thymine is not there in RNA. So what will come? Uracil will come. There is G over here, so C will come. C over there, so G will come. The body looked at this one and be like, okay, fine, let's create an RNA. So from the DNA stage, an RNA gets created. Now, this messenger RNA, I told you it's a messenger. Where does this take the message to? This messenger RNA, which is right now UCG, will go to another cell organelle called as your ribosome. Ribosome. Once it goes to the ribosome, this is a cell organelle inside the cell. Okay, so inside this ribosome, once they get this UCG, they'll be like, UCG. Okay, they're like, okay, this means something. And then what they will do is, once that comes over here, they will not, because it's the complementary thing that has come, right? So there, this messenger RNA, once it comes to the ribosome will be like, they send UCG. Which means ideally what I have to create was what? Instead of U, what should be the pair that comes? A. Instead of C, what is the pair that comes? G. Instead of G, what's the pair that comes? A, G, C. So in this manner, we've got a thing into the ribosome, and the ribosome will be like A, G, C. Okay, UCG did not make much sense to the ribosome. UCG came, and then ribosome like UCG would mean AGC, and then the ribosome will create a protein. Once that protein is created, when that protein is expressed in my body, then I can say that gene got expressed in my body. Which means I would have had the gene for black eye. Now I get it expressed when that gene turns into an RNA, that RNA turns into a protein, and that protein is expressed in my, just having that gene will not just give it to me. That gene has to get converted into an RNA, and the RNA has get to convert it into a protein, and when the protein is there is where I understand that, or when that protein is expressed, I know, okay, I have that feature. So when I'm saying a gene is functional, I mean to say a gene is something that is capable of creating a protein. I hope this much is clear. If this is clear, let's make it into a very simple process. DNA in your body turns into RNA and RNA turns into protein. The process by which DNA turns into RNA is called as transcription, and the process by which RNA turns into protein is called as translation. Once a gene is there, it has to undergo transcription and translation and create a protein. And when that protein is expressed, I know I have that feature. Take a moment for this, and this is the base of all biotech that we're going to study. Why? Because I just told you there is DNA, then there's RNA, from the RNA stage there is protein. And when the protein is expressed, when the protein is created, and at sometimes even after the protein is created, sometimes it's not expressed fully. So all that process has to be completed for you to have a feature. Which means if you have black hair, it's because of this. If you have short height, it's because of this. If you have cancer, it's because of this. If you have any disease, it's because of this. Why? Because you just did not have a gene. The gene got turned into an RNA, into a protein. Every single feature you have, which is of your body, is because this process has happened. Which means if I want to change any feature of my body, should I not start tampering with this process itself? Because there's nothing else I can do. Right? What all can you change? So let's have a look at this. And this is one of the most interesting parts that I like to teach students. So I told you DNA is there. DNA gets converted into RNA, and RNA gets converted into protein. Okay, let's take our example itself. So there was a DNA part which was your ACG. Maybe I can start doing at the first stage itself. At the first stage itself, ACG is there. I don't want that feature. Whatever features ACG is giving, I don't want that feature at all. So what I can do? I can just remove ACG. I can delete or I can edit. Delete something means what? I remove ACG completely. Edit means what? ACG is the code. So what if I change G into C? It got changed. It's ACC now. What if along with this ACG, I am putting another C also? I'm adding something. I'm adding something and suddenly the body is like, ACG is for black hair, but ACGC is for glowy skin. ACGC is something else altogether. So if I know that, I can just add something, which means I can start tampering with the DNA itself. I can delete or edit the DNA. I can delete or edit the entire code. I can edit the base pair. Base pair means what? Just this G. I don't want the G. I can remove it. I can change it into another C. I can edit that. Okay. All of that I can do. Now I am like, okay, this ACG is pretty useless. But instead of ACG, if I had GCG, That would have been nice. Maybe some extra feature. Now I'm seeing GCG is there in another organism. Okay. So I'm like, hey, you have GCG. I could just take that GCG from there and I can put it over here. Right? I can create something. I can add something into my genetic material. Okay. Now suppose these are all the remember this. So whenever we study biotechnology, we are actually sitting and tampering with each of this. I am either changing the base pair or I am changing the code. I am adding something to the code. I am deleting something from the code. I am removing the code completely. Any of these things I can do to the DNA. Now suppose I am like, okay, fine, work on the DNA is too much. How will I find this code in this entire body? So I am wait. Anyway, one day, not one day, every day it's going to happen. The DNA which is useful or that code, it will uncoil and will create what? An RNA. An RNA is easier to find maybe. So I wait for the body to create the RNA. Now, once the RNA is created, think about it. I told you once the RNA is created, the messenger RNA goes to the ribosome. Once it reaches the ribosome, only the translation process starts. What all can I do over here? First of all, when the RNA is created itself, messenger RNA is created itself, I can destroy the messenger RNA. If I destroy the messenger RNA, will that messenger RNA ever reach to the ribosome? No. So that protein will never get created. But what's the problem? I have to, each day this is happening, no? So which means that each day I have to wait and I'll be like, okay, fine, kill the messenger RNA, delete the messenger RNA. I have to keep doing this. That's quite difficult. What else can I do? Okay, we let the RNA get created. We let the RNA get created, but it's like this kidnapping part. The RNA, messenger RNA, never reaches the ribosome. it's like some plot. What has happened? RNA gets created. I'm not destroying it. RNA gets created. But the RNA does not reach to the ribosome at all. Somewhere along the path, RNA is gone. Poof. I have silenced the RNA. I can silence the RNA. If it doesn't reach the ribosome, the rest of the process will not happen. So, I'm not destroying it. I'm adding something into my cell, which is like the messenger RNA will come out of this nucleus, and the messenger RNA is going to go to the ribosome and suddenly gone. What has happened over here? I can use certain external substances and I can prevent the movement of the RNA to the ribosome. Okay. If I'm not doing that also, what is the next process? Okay, messenger RNA got created. I am not destroying it. I am not silencing it. What am I going to do? I can do this tampering on the messenger RNA also. So as I saw over here, ACG got created. Instead of that, I got a UGC. UGC is going to go to the ribosome. Halfway through the part, I am like, messenger RNA, just 1 minute, come over here. And instead of you, I am replacing it with something else. I can do that also. I can edit the messenger RNA. This is not a very advised process. Why? Because the last one. Okay. Why? Because in the first point itself, once this messenger RNA is a very flimsy creature. Okay. It's like messenger RNA is like, I just want to give my message and go. Okay. So it's not a very stable component. So to do research on the stable component can be a bit difficult. That is why editing of messenger RNA is not generally preferred, but the other two processes can be easily done. Now finally, we are like, okay, fine, somehow we didn't do anything to the DNA. We didn't do anything to the RNA. So RNA has gone and created protein. Then what is the last stage you can do? you can somehow avoid the expression of protein. Now I want you to take a look at all of this. Every single biotechnology related process that we are going to study is one or the other of these processes. Got it? Either you're deleting or changing the DNA, or you're silencing the RNA, or you're avoiding the expression of the protein. So understand, you are either tampering with the transcription process or the translation process. Are you getting this clear? Because I want you to understand the possibility that you can change any of these things. For non-science students, when I teach this topic in class, I get, ma'am, can we change that? Can we change? Yes, you can change anything, because these codes which are there in the body, you just have to find where that code is or which is that code. Once you get the code, you can change any of the. Science has developed to such a manner where you can delete, edit, add, insert, silence. All of these processes can be done, and this is the basis of biotechnology. Okay, now a pointer to be noted over here. I want you to keep this also point in mind. This is just a casual thing. If you've heard, some mushrooms and everything which is toxic in nature, what happens in the case of, when you consume toxic mushrooms is, these toxins go and interfere with this process. Okay, so you have the DNA has to get converted into the RNA, right? For all the features, your breathe, everything in your body is determined by these codes. So what happens is, the DNA is created, but at that point, if you have had toxic mushrooms, they stop the creation of mRNA, which means what? And if you're continuously consuming the toxins, what happens is mRNA is not created. If mRNA is not created, protein is not created. So this is the reason why toxic mushrooms can be fatal, because all of your body features are based on the creation of, are based on all of these processes. You need to have DNA, you need to have the mRNA and the protein which is related. So any of these processes get stopped unwantedly, that can be fatal for us. Got it? So that is the reason why mushrooms are considered toxic. Anyway, coming from all of that, the next thing that I want to teach you is, this topic which is called as recombinant DNA technology. Primarily, understand that now you know very clearly what this is, because I told you transcription, translation, everything. So recombinant DNA technology will be what? You're doing something to the DNA. What are you doing to the DNA? Here what happens is, now there is some part of the DNA of one organism that I find is very good. Okay, now for example, let's take the mustard plant, or maybe the cotton plant. Okay, the cotton plant in India has very good yield and everything, but I also want the cotton plant to be very resistant against certain insects. Okay, so what I do is, I look for any other genetic code which expresses, not in the cotton plant, in any other organism which is showing that kind of a fighting mentality to these insects. And I get the code in one organism. It might be a bacteria. I only have to look like, okay, is whatever that organism, is it having the same genetic material as that of the cotton plant? So which means genetic material means what? DNA, DNA, RNA, RNA. So I'm looking and I'm finding this very nice bacteria which has the ability to attack all of these insects. Bacteria has DNA as its genetic material. So I'm like, okay, bacteria, I start looking into the genetic code of the bacteria, and I understand that the bacteria is able to behave in this fighting mentality to the insects because it has a genetic code. I look at what that genetic code is. And once I get that genetic code, I'm like, see, so I have this genetic code, but that genetic code being in the bacteria, it's not good for it's not actually doing any good for the cotton plant. I somehow have to get that genetic material into the cotton plant. So what do? I cut or isolate a segment of the DNA or the desired gene. I cut it from there, and by cutting it, I put it into, and I come over here and I paste it, or what do I do is I join or transfer this DNA or gene with the DNA of a different organism, which means I can cut it from the bacteria and I can paste it into the cotton plant. Very simple, no? Yeah, but it's not as simple as this. Understand, here what happens is, this recombinant DNA technology has 3 major stages. I am cutting the DNA using an enzyme called as restriction enzyme. Okay, once I have cut the desired part, let's say I have cut the desired A, C, T, I have cut that desired gene. Now I have to put it inside the cotton plant. I cannot be like, okay, fine, cotton plant, let's go stick it over there. That's not what happens. What I am doing is, this cut ACT thing, I will place into something called as a plasmid. Okay, what is a plasmid? A plasmid is basically a circular DNA vector. When I explain this in class, I just tell students like, we have courier service, right? The function, like for example, I am sending a few books from one place to another. The courier service is just a medium. The courier service will not open the book and write into it or anything, right? Courier service like, I'll take it from here, I'll go and transfer. So the plasmid is actually something like that. It's a vector carrier which is circular DNA in form, so it's easier to attach this part. So what happens is, I take this circular DNA, it can be again, this vector can be artificially created, it can also be taken from other organism. Okay, you look at the plasmid, you see if it's not having any infectious trait or anything, you can take it from another organism, or you can artificially create this courier service in your lab. Whatever you do, what you do is, you cut a part of this, so this part is gone. Okay, now into this part, you place this ACT. When I am placing it, I will stick it with this enzyme called as ligase. So you use another enzyme called as ligase to join the DNA with the plasmid. Once it's created in this manner, so now the plasmid has that added ACT. I create several copies of it, several copies of it in the lab. And once I create several copies of it in the lab, I put it inside my host organism, which in this case is your cotton plant. What we have done right now over here is recombinant DNA technology. I have taken something desirable from one organism and I have done this. I've joined it with something else and I've sent it into the host organism. Okay. The next important, so far we are covering the important technologies which is there. The next important technology that you need to know in biotech after your recombinant DNA technology is something called as cloning. Okay, to understand cloning, again cloning is in news this time because the founder of the cloning process and everything, founder in the sense that he was the one who experimentally proved it, he had passed away this year. So there's a lot of, and there's also an anniversary year of cloning, etc. So just have a look about it. First of all, what is cloning? A lot of people get quite confused when it comes to this. See, the idea of cloning is to create genetically similar copies. Okay, fundamentally the idea is to create genetically similar copies. How do you create something that is genetically similar? Okay. Now, here is where you have to understand the very process. Now, when I'm saying cloning, you can have different types of cloning. You have therapeutic cloning, reproductive cloning, etc. Whatever is the type of cloning, the first step of any cloning process is something called as somatic cell nuclear transfer. Now, you all know what somatic cell is, you know what nucleus is. Now, let's look at the process. I want you to pay attention over here. So, what you are trying to do over here is, you take a somatic body cell. You take the egg cell. Okay, can be from the same organism. It can be from different organism. Whatever it is, you are taking a somatic cell, taking the egg cell. Now, at each process, I want you to tell me what am I doing over here. I take the somatic cell and I am removing the nucleus of the somatic cell. Okay. I take the egg cell. Here also I am removing the nucleus, but I am not using the nucleus anymore. I am throwing away the nucleus. Okay. At the end, what I am having with me is a nucleus that I have taken from the somatic cell and the outer body that I have taken from the egg cell. Egg cell is what? A germ cell. And I put it both together. So, this cell which is having right now, it has the body of an egg cell and the nucleus of a somatic cell. Why did I do this? Or what am I trying to achieve with this process? What exactly is cloning? Now, here look at it. I want to create a clone of myself. Okay. At the current natural human reproduction process, what is the factor that is holding back complete clone of a creature? It's the fact that in the current human reproduction or animal reproduction, a fetus is created by the germ cell from both the father and the mother, which means both of their features will be present. Okay. So now I don't want, if the father is there, the father wants just a clone of him, or the mother is there, the mother wants just a clone of her. So in this case, how do you do it? So if the mother wants a clone of just her, only her genetic material has to get passed on, right? How will her complete genetic material get passed on? Now, I initially told you the somatic cell has 23 pairs of chromosome. The egg cell just has 23 chromosomes, which means if the mother wants a complete copy of herself, all the 23 pairs should go to the next generation, not just 123. So here what has happened is, when I am taking the nucleus out of the somatic body cell, I am getting 23 pairs of chromosome from this. Okay. Then in that case, why did I remove this? If I did not remove this nucleus, what is going to happen? I get 23 pairs from the somatic cell. Then in the egg cell, there will be just 23. In total, there will be 23 pair plus just one more 23 chromosome. That is not even going to create a human baby. So, you have to remove the nucleus. So, I am removing the nucleus. Then why am I not doing this just with the, I could have just taken the somatic cell and created copies of it, no? Why am I not doing that? I'm not doing that because if I need to create a future generation, the correct, you remember all the gene and everything related to that, that will be there in my germ cell. That will be there in my egg cell. So, if I am just taking somatic cell and I am creating copies of it, I will just get copies of my somatic cell. But if I want all the genetic material of my somatic cell to be created into a future progeny, I have to use a germ cell because your germ cell is reproductive in nature. So, what happens is, so I take the germ cell and I put it over here, and then what am I getting? I can, in this form, I can create several copies of it. Now, this is where it gets interesting. So, I told you this is the beginning of all cloning process. Okay. So first I do somatic cell nuclear transfer. Once I do somatic cell nuclear transfer and I create this set of cells over here, I can create this in the lab, and from the lab I can place this into a uterus. Now my question is, does this have to be the uterus of the mother itself? Not necessary. You can place it into a surrogate also, and you can create a baby which will be genetically similar to whoever contributed your genetic material of the somatic cell. So let me ask you something over here. Using this process, you can create clones of both males and females, right? If a male is giving the genetic material, his clone can be created. If a female is giving, his clone can be created. Okay, and I also want you to remember, each of these processes over here is something that can happen in the lab, and finally the surrogate will give birth. But surrogates, none of its genetic material is also going to be there. Now, if this is the process that is done, it's called as reproductive cloning. You have created the clone of yourself or whatever is the creature using this process. But instead of that, okay, I told you there is another process called as therapeutic cloning. See, can you see this set of cells over here? Technically, what has happened over here? In our body, I told you, there will be a single cell zygote. The zygote will start multiplying into different cells, into number of cells, 16 cells, 32 cells. At that point, what is there or what is the feature of it? It will be very differentiating in its nature. Naturally, this is happening. The same thing can happen in the lab conditions also, which means what has happened over here? I have taken an egg cell and I am creating several copies of that egg cell which has this new genetic material, and here this new genetic material thing which is over there, I am giving it certain growth conditions. This cell will rapidly start multiplying. It will be in a stage of 16 to 32 cell stage. At that point, it will have the feature of pluripotency. When it has the feature of that pluripotency, can I not take those stem cells, and if someone is sick or someone is having some features, or if there is a correct match in this manner, can I not create some, for example, someone has lost a part of the liver, I want to create a new liver for them. So can I not take that stem cell which is very highly pluripotent and create an organ for them because it's pluripotent, right? I can actually create a therapeutic clone for that sick person. So understand, initially when we start somatic cell nuclear transfer, you do this process, but at the end, if you're putting it into a surrogate, it will become reproductive cloning. If you're allowing it to multiply and create stem cells, and that stem cells is used for some disease recovery, that is called as therapeutic cloning. Got it? So this is the reason why it was mentioned. Both of these are the beginning stages, but what happens after that is dependent on what you're going to do with this particular clone. Okay, so a few pointers before I go into the next topic. Do we have any natural cloning process without all of this? Yes, you do have natural cloning process in a lot of plants which rely on vegetative propagation. You do see natural clones. Even in humans itself, you do see when you have twins that are formed from the same zygote. Why am I specifying this? Sometimes in humans, twins are formed because there are two egg cells. Okay, if there are 2 egg cells, 2 sperms will come and fuse, and then you will have 2 fetuses. They need not be clones, because the fusing and everything, they'll create absolutely unidentical copies. But sometimes after a sperm has come and fused with the egg and a zygote is created, the zygote splits into 2. And the zygote, when it splits into 2 and creates twins, at that point, they happen to be clones of each other. That is where you get your identical twins. Okay, so that is also close to a cloning process. Now comes the important technology that we have to deal with for this year, primarily because this year, for this particular technology in UK, they have given, or they've said that they've created the first, see, until now it was a very theoretical thing. You've had it happen in Japan or some, in some places, but it was not like a general method that was used to treat, to create a baby or anything. But very recently in UK, they've created a 3-parent baby. And UK is now planning to license this operations of this creating a 3-parent baby. So again, it has been in news. Let's have a look at what this idea is. Now, at this point, just to recall back into something that I taught you. I taught you the genetic material of a baby or anything. Okay, comes from the mother and the father. Now, let's see who has more stake in it. Why? Because the mother has a egg cell which is placed in her body. The sperm comes from outside. Now, let's look at where all the genetic material is. In the egg cell, there is a nucleus which has the mother's genetic material. I also told you in the humans, there is the mitochondria which has the mother's. And if it's the mother's mitochondria, I mean, it's not if it is the mother's mitochondria, it will be the mother's mitochondria over here. Some genetic material will be there also. When the sperm cell comes, what primarily happens is, the sperm cell reaches the egg, the nucleus of the sperm cell merges with the nucleus of the mother's cell. Okay, it's the nucleus which is merging. No other cell. It's not that the sperm will come and directly it will hit and all the organelles will be doubled. Nothing like that. Just the nucleus will come and fuse with the mother's nucleus. So the point is, ideally in a human baby, the human baby has the mother's nuclear genetic material, the father's nuclear genetic material, and the mother's mitochondrial genetic material. This is what is present in you. 3 things. Now, this is where the catch is. Sometimes the mitochondrial genetic material, if it is having some defect, some genetic defect or anything, it will get passed on. Who is having the mitochondrial defect? If the father has the mitochondrial defect, that will not get passed on in this manner. Or it is not. As of now, there are no proven ways where it is getting passed on. Why? Because the father's nucleus is only fusing with the mother. The father's rest cell organelles do not have a role over here. Understand? So, if in an ideal human scenario, the baby has mother's nuclear genetic material, father's nuclear genetic material, and mother's mitochondrial genetic material. Now the problem is, if the mother's mitochondrial genetic material is defective, what happens? That defect will be carried on to the next generation. And this is where we need to somehow prevent that mother's mitochondrial defect from moving on to the next generation. So here is where what happens is, you can do it in 2 ways, 2 methods. First thing is, I let the fusion happen, as in there is a defective mitochondria of the mother, there is the healthy egg cell, both of them fuse. Resultant cell will have what? Resultant cell will have a nucleus which has mother's genetic material, father's genetic material, and a defective mitochondrial genetic material. Okay. At this stage, after the fusion has happened, what I will do is, I will remove the defective mitochondria. I will remove the defective mitochondria, and then I will place I'll get a donor, and the donor's mitochondria will be placed. And mind you, because this is all done in an egg cell, the donor is highly probable to be a female, because the mitochondria, you have, in science you always try to avoid all cases of mismatches that is there. So you try to get the mitochondria from another egg cell and you place it into the mother, or this cell which is, you remove the defective one and you place the healthy one. Now what does this fetus have? Mother's and father's nuclear genetic material, donor's mitochondrial genetic material. Okay. Now it's a zygote, and the zygote will start developing. So 99% of the genetic material is from the parents. One small minuscule percentage is from the donor. And because all of them have contributed to the genetic material, it's called as a 3-parent baby. Now, this is one method I told you. I have allowed the fusion happen and I have removed the mitochondria. I need not do that also. In the initial stage itself, when the mother's egg cell is found to have a defective mitochondria and the father is having a healthy nuclear material and everything, at this initial stage, before the fusion or before the fertilization itself, I can remove this defective mitochondria outside. I can place the donor's healthy mitochondria inside the mother's egg cell and then let them have the fusion. When they have the fusion like that, what will it result in? Again, a zygote which has a donor's mitochondria and mother's and father's genetic material. So what I mean to say is, I can create a 3-parent baby before fertilization and after fertilization also. Okay. The point is, I remove the defective mitochondrial DNA of the mother and I substitute it with the donor mitochondrial DNA. I hope this technology is clear. Okay. And I want you to keep this in mind. A lot of students keep asking me, is the mother's genetic material only the reason? Yes, it's only the reason. At this particular point, I hope now you will not ask such a question, because I hope this is clear to you. Why it is the mother's only thing? Because the fusion is happening at the site of the egg cell. And the fusion is happening between the nucleus of both these cells. Not, and when you have the, what you say, father's entire sperms, rest all organelles, they are not fused with the mother's egg cell. So whatever is the site of fertilization, there is a defect, and this is the defect that is there. That is the reason why it is the mother's mitochondrial defect which is corrected using the 3-parent baby. Another term for this is also mitochondrial therapy. When you hear mitochondrial therapy, don't be like it's some new process, it's the same thing. Okay. So I hope this is also clear. So I think we've done quite a few technologies at this point. Next thing that I want to teach you is, again, something that's doing its rounds, very small topic, genome sequencing. Until now, I have told you that there is genetic material in the DNA, in the RNA, in the chloroplast, in the mitochondria, etc. So now, what you're going to look is, you're going to figure out what is the order in which all these base pairs are placed in. Why are you doing it? So that you know what genetic code is contributing to what feature. Okay. So genome sequencing basically means trying to figure out the exact order of base pairs in an individual. You can do that using saliva, epithelial cells, you can take bone marrow, hair, seeds, any of these things from the body, and you can try to sequence. You don't need a full human sitting all of the time over there. You can just collect the genetic material and you can keep sequencing and figure out what is the order in which the base pairs are arranged. Okay? Now I want you to understand, based on this genome sequencing technique, that there is a current affair that has happened this year. It's called as Infexen, INFEXN-TN. What is happening over here? It's India's first genomics-based infectious disease test. Okay. What are you going to do over here? Here, you are, whenever there is any disease or any causative organism out there, this technology will help you to figure out the genome sequence of that disease causing agent within 24 hours. Okay, so you're going to figure out the correct genome sequence of, for example, Nipah is spreading. You do not know what variant it is. Get the sample of Nipah. Using Infexen TM, you will be able to figure out the complete genome sequence of it quickly, so that you know what is the effect it's causing onto humans. How can you prevent the spread? If you want to create a vaccine, you can do it. If you want to create a medicine, you can do it. All of these things can be done in a quick manner. So Infexen TM, that is developed by IIT Bombay based Haystack Analytics, and NIMS Medicity, which is there in Kerala. They both have come up with this technology to, it's India's first genomics-based infectious diseases test to detect any bacterial or fungal infection, along with detection of any antimicrobial resistance genes. When you are genome sequencing, you will figure out all of these things, right? The catch, or the important thing of it, it is all happening in 24 hours. You are using this to detect bacterial and fungal infection. Why am I specifying it? You will understand that viruses undergo mutation at a very, very higher speed. So what happens is, it might not be much effective when it comes to viral infections. That is why it's specified bacterial and fungal infection. I hope this is clear. This is the current affair I wanted you to deal with over here. The next topic that I want to deal with you is something called as synthetic human embryo. The reason I put this is because I want you to keep this in mind. They can ask you a very tricky question based on this. For the very first time in Israel, scientists have now grown a human embryo in the lab without using an egg or a sperm. Okay, without using an egg or a sperm. How did that happen exactly? What they've done is, instead of using a human egg or a human sperm, they've taken a stem cell of a mouse. Okay, and in the lab, they've created experiments enough to start forming when the stem cell was taken from the mouse. So that's also not human. Okay, it was taken from the mouse, and in the mouse's stem cells, they kept on giving different mediums and everything. And very soon, the mouse's stem cells started behaving like human stem cells. And you know what human stem cells are pluripotent. Even the mouse's stem cells are pluripotent. That is why it was made to behave like human stem cells. But imagine, from the mouse's stem cells, they started behaving like, it started mimicking an embryo, and very soon, from the mouse's stem cells, they created cells which seem familiar to a human fetus. And then the human fetus started developing with hands and limbs and everything like humans. So, what is your takeaway from this? Now, here is where your stem cell topic gets completion over here. We talked about, till now, we were all talking about what? You want a human organ, you take human stem cell. Now, the possibility is, you need not even take a human stem cell. You just take a stem cell from any other animal organism. and you can recreate it. So that is your first takeaway. You need not, when I want to do stem cell therapy, the possibility is that I can take the stem cell from mouse or cow or any of the other organisms based on the match, and I can make it to behave like a human cell. Okay, that's the first thing. Second important takeaway is that they've created an embryo without an egg or a sperm. So that itself is the second takeaway, where they ask you a tricky question based on it. Yes, you can create. Now with the technology happening, you can create this also. Okay, so I hope all that part is clear. See, now there might be a question as to why we've not discussed CRISPR-Cas9. See, CRISPR-Cas9, again, very simple terms you have to understand. I told you that you can tamper with the DNA, you can tamper with the, any of your genetic material by cutting things. Now understand, CRISPR-Cas9 is a very efficient scissor, genetic scissor which is there. Which means whenever someone asks, can I do recombinant DNA technology using CRISPR-Cas9? Yes, you can do it. Can I do any of your, any other kind of technology where you have to cut genetic material? You can use CRISPR-Cas9. Understand? So the level of questions asked for CRISPR-Cas9 is already exhausted. This is the reason why we are not going deep into it. But there is another technology called as CAR T-cell therapy and everything that we will be discussing in the next section, which is health and diseases. As you know, there's a lot of diseases that keep doing its round every year, but very less questions get asked from this. One area that I do feel is important with respect to this year, I'll just quickly deal with you, and this is an area with a lot of students do have a doubt also. So let's have a quick idea of it. Yeah. See, now when it comes to human health and diseases, you have a major thing called as your pathogens. Pathogens are your disease causing agents. You have virus, bacteria, fungi, protozoans, any of these are your disease causing agents. Now also understand the fact that out of all this, virus is much more sought after one, because the nature of viruses are quite different. I'll tell you why. Virus on a normal scale is a very crystallized. It might be all over here, all around the surroundings. It will be crystallized in a manner. But the moment virus comes in contact with a growth substrate, with a living substrate, it will, start being alive, to start replicating. Okay, so this is a quite different feature, and also the feature that virus has this feature where it constantly tries, for virus will have its own genetic material, can be DNA, RNA, whatever its genetic material. Virus has this feature where once it enters into the body, it tries to go, and if it is possible, if it's a prolonged infection or anything, it will try to somehow go and attack our genetic material. When I'm saying attack our genetic material, it's very, very interesting how it does it. Now, for example, virus, which is having DNA as its genetic material, enters into our body, looks at our genetic material, sees like, okay, DNA. Okay, fine. After some point, what happens is, slowly the virus will start affecting the cells, and at what stage, if it's the same genetic material, what happens is, it might, instead of the cell's DNA, the cell's DNA gets replaced with the virus's DNA. So then, will the cell behave like a human cell or like a virus? It will start behaving like a virus, because it has now the genetic material of the virus. So then our own cell will start behaving like a virus, and it will start replicating. So this is also the reason why viruses are studied in depth. And just a pointer over here, viruses' genetic material can be DNA or it can be RNA. When the virus's genetic material is DNA, it can be a normal double-stranded DNA, like our normal DNA, or it can be a single-stranded DNA. So what do you mean by single-stranded DNA? It will be one strand, and the important factor is it will have thymine. It will not look like an RNA. It will have thymine instead of, in RNA when it's single strand you have uracil, right? But no, this is DNA which has become a single strand. Similarly in RNA also you have double-stranded RNA and normal single-stranded RNA. So viruses can be of any of these genetic material. When I'm saying double-stranded DNA, your pox virus or your common cold causing adenovirus, all of them are in this category. When it comes to single-stranded DNA, certain viruses like parvovirus are in this category. When it comes to double-stranded RNA, important virus called as rotavirus, which causes diseases in India. You have even a vaccine called as Rotavac against it. All of that is caused by your rotavirus. Single-stranded RNA, you have retrovirus and rabies. Retrovirus is the one that happens in HIV, etc. Okay, so if this and your coronavirus which was there, it had your RNA, normal single-stranded RNA as its genetic material. So understand, now in the case of pox virus or adenovirus which causes common cold, etc., once it enters the body itself, you start having symptoms. So what happens is you try to stop it then and there. This is the reason why, despite it having very similar structure to our genetic material, it does not have enough time or enough infectious ability to go and start swapping our genetic material. But in the case of certain viruses like your retrovirus, rabies, etc., what is happening over here? You can see over here, it's a single-stranded RNA. Important thing to understand over here is, so this retrovirus enters into our body. I earlier told you this about HIV. It enters into our body. It's like, okay, I am single-stranded RNA. Body is having DNA. Okay. So there's no match at all, okay, to affect it. It's at that point, what happens is, in this retrovirus, a certain enzyme called as reverse transcriptase. Reverse transcriptase gets generated. Reverse transcriptase, if transcriptase is converting DNA to RNA, reverse transcriptase will do what? It will convert RNA to DNA. So suddenly this retrovirus which is over there, which came into our body with just single-stranded RNA, with this enzyme called as reverse transcriptase, which is in its body itself, it has now changed its genetic material to DNA. And once its genetic material is changed into DNA, then what is the problem? Our genetic material is also DNA. So then it will affect it. You do see this happens in the case of a very prolonged infection. Okay, so this is one area that needs clarity over here. I hope that is clear. The next thing that I want you to have a little bit clarity, and it's also a current affair, is, as I told you, virus is always in news. This year, they have, around in February time, I believe, they've discovered a new organism called as obelisk. Okay, now what is obelisk? I want you to understand, there is something called as your virus, and there is an organism much smaller than the virus, which is called as a viroid. Okay, Now, obelisks are an intermediate kind of organism. It has both the features of virus and viroids, and that is an obelisk. At this point, I'm pretty sure they will not ask you a question on obelisk, but they can ask you a question on virus and viroid. So let's have a quick idea about what is their differences. A virus can have RNA or DNA as its genetic material, as I just told you. But in the case of a viroid, the viroid just has a RNA as its genetic material. In the case of a virus, outside the body of the virus has a protein covering, protein coat. In the case of the viroid, no protein coat. Okay. When it comes to the virus part again, it can infect your plants, animals, or even other microorganisms like bacteria. But in the case of viroids, they affect only plants. Okay, so I just wanted you to have this differentiation also. So obelisk is considered to have both of these features. So obelisk has both RNA and DNA as its genetic material. Some features have protein coat, some don't have protein coat. So they are like this middle path thing which is there between virus and viroid. So I don't see much question opportunity for obelisk, but for virus and viroid, I hope this part is clear. Okay, so this is one thing that we need to get clarified in your health and diseases. Another thing that comes important as a part of your health and diseases is your vaccines. And here, one of the things that we have to discuss with much urgency is not your conventional vaccine. I hope you all know what is the idea of vaccine. The idea of vaccine is the very fact that you are training your body for identifying some kind of a disease-causing agent. Okay, so the disease-causing agent can be a bacteria, virus, whatever it is. So you are familiarizing your body. It's like showing the body a picture and saying like, hey, look at this. If this one comes, you attack it. Now, instead of the picture, what all can you show to the body? You can maybe take the virus itself or that bacteria itself. You can maybe weaken it. You can make it in a very weak form and show, see, this is what it looks like, you attack it. So all your conventional vaccines which are there, live attenuated vaccines, non-live vaccines, toxoid vaccines, subunit vaccines, conjugate vaccines, all of these vaccines, what they do is they try to train the body using some part of the virus or the bacteria. Here, live attenuated, you take the virus which is causing the disease and you weaken it. It's not fully dead, it's weakened. In the case of the non-live vaccine, you take the virus and you crystallize it. Crystallize it means what now? You are not giving any growth medium, you are making the virus dead. But is the virus actually dead? I just told you, around us also the virus is crystallized. Okay, which means what? Given the right conditions, the virus can be activated. So there is a risk involved in it. So in the non-live one, the virus is crystallized. In the toxoid vaccine, what happens is the virus might be releasing some poison or toxins. I take a part of the toxin and I familiarize the body with that toxin that the virus is releasing. In subunit vaccine, maybe the virus has some kind of a protein covering or something. So I take part of that and that is given to the body as vaccine. So understand, in all of these things, you're taking some part and you're familiarizing. Now, the point is, there's always a question of efficiency. Okay, what if the virus becomes activated? What if the body is not trained enough? What if the body does not identify? So this is where there is a couple of new age vaccines, and these new age vaccines are not going at the outer covering or anything. Now we know very clearly what makes us is what? Our genetic material. Okay, even for the virus, it's like that. Virus might have outer covering and everything, but at the end of the day, the essence of the virus is where? In its genetic material. So now instead of just showing other parts, the scientists are now using the genetic material of the disease causing agent and making that into a vaccine and inserting that into our body so that the disease cause, that genetic material, our body is like, oh fine, this genetic material, the virus can come with a new hairdo and a new protein coat and everything, but the genetic material remains the same. The infectious genetic material remains the same. So this is what is happening in the case of DNA vaccines and mRNA vaccines, etc. In the case of DNA vaccines, as obvious, what is happening is I can take the genetic material of the virus, or I can also do one more thing. I can look at the genetic material of the virus and I can create that genetic material in the lab. Now you know we can create the DNA slices in the lab and then I can create a vaccine. So my point being, when I'm creating DNA vaccine, the DNA can be directly taken from the disease causing agent, or I can also use genetically engineered DNA molecules and I can create the vaccines. The rest all thing remains the same, guys. Nothing extra. You are creating a vaccine using the genetic material. I can either take the genetic material directly, or I can manufacture the genetic material. Same thing comes for your mRNA molecules also. What happens over here is, in the case of this, as I told you, whatever is the genetic material of any creature, if there is a DNA in it, for example, there is a virus out there, okay? The virus, it's having a DNA. Even the virus's features will be expressed when it has a mRNA created and the mRNA goes there and everything. So if I can create a vaccine by recreating that mRNA, isn't it much more specific than the DNA? So again, what am I doing is, I'm looking at the disease causing agent, I'm looking at what is the mRNA of it, and I'm creating a vaccine. This is considered to be much more effective than DNA vaccines because mRNA is created after a further step. Any errors in the DNA are all solved and mRNA is created only very specific genetic compounds. So when I'm creating a vaccine using mRNA, it will indicate only and only to that specific virus. mRNA is a very popular technology. As you know, mRNA technology is a Nobel Prize winning technology at this point. Why? Because of its specificity. Okay. And the next important thing that I want to discuss in health and diseases is a current affair based thing. So before I go into this current affair, I want you to understand that we have a new vaccine that's doing its round. It's called as Servavac. Okay. Servavac is India's first quadrivalent human papilloma virus vaccine. So human papilloma virus is a kind of virus that can cause cervical cancer in women, and very recently Servavac was created as a vaccine against this particular disease. It's called quadrivalent human papilloma virus because human papilloma virus has different strains, just like COVID had different strains, etc. So quadrivalent means it's against 4 strains. Against 4 strains of your human papilloma virus. It's created like a hepatitis B vaccine which is there, and it was created by Serum Institute of India, Department of Biotech and the Biotech Industry Research, BIRAC, which is there. This is one current affair associated with this area. The next current affair is HaviSure. HaviSure is India's first indigenously developed hepatitis A vaccine. At this point, we have to look a little bit into hepatitis because that is a disease that has been recurrently even asked. I want you to understand hepatitis can be of 5 variants. A, B, C, D and E. Out of all of these, hepatitis B has vaccination. Hepatitis A currently has a vaccination now. Hepatitis C does not have a vaccine. And hepatitis D and E are generally considered to be, D is a much more severe case and E is a much more lighter case where it comes out to be waterborne. So the vaccination is not much of a pointer over here. Okay, now here I have to understand that HaviSure, which is there, created by, important thing to be noted, India's first indigenously developed hepatitis A vaccine. It's recommended for routine immunization in children. It is administered in 2 doses. The first dose is given at about around 12 months of age, it's 1 year, with the second dose at least 6 months after the first. All this is clear. Now the catch is, this vaccine was developed by Indian Immunologicals Limited, which is a subsidiary of National Dairy Development Board. It's not something that you associate with human vaccines generally, but National Dairy Development Board is the mother organization. IIL is the the subsidiary under it, and they have developed HaviSure. So, point to be noted. Moving quickly into areas of space technology and defense, we have few current affairs that we have to look in this. One of the first important current affairs that we have to look in this field is the sun missions that are currently there. There are currently 2 solar missions which are pitched for learning the sun, and here is where you have to understand these missions are, I hope the geography part is covered, that the sun structuring and everything. So you do know that the sun's certain areas, ideally if you have the sun's area and the atmosphere around the sun, we expect that the area very close to the sun will be much more hotter than the area above it. But it's not like that. What has happened over here? The area over here is relatively, when I'm saying colder, understand it is not freezing cold or anything. The temperature is less. That is what the meaning of it. It's still Kelvin high. So, relatively colder than the area around it. Okay. So, now there's a lot of interest into why this is happening, and to understand that, so the comparison over here is between the Aditya L1 mission and the Parker Solar Probe. Aditya L1 mission by ISRO, Parker Solar Probe by NASA. Aditya L1 mission will be at a distance of almost 148.5 million kilometers from the sun. Parker Solar Probe will be at 6.2 million kilometers from the sun, which means that Parker Solar Probe will be much more nearer to the sun. The next important point about the Parker Solar Probe is this: it will be the first spacecraft to fly through the sun's corona. Corona means what? The outer atmosphere region of the sun. Aditya L1 mission is not going anywhere close to the sun. It is situated at the Lagrange point 1. I hope you all know what the Lagrange point is. So understand that it is away from the sun, and it's an observation mission which is there. Again, because it is an observation mission, it will need much more instruments to sit and study and everything. So here you will understand that Aditya L1 mission has 7 instruments that it's carrying along with it, and Parker Solar Probe has just 4 instruments that it is carrying with it. Okay. The next important thing that I want to discuss with you is obviously about the Chandrayaan 3 mission which is there. When it comes to Chandrayaan 3, I do think the question will be a very comparative mission between 2 and 3 and 1. So, first of all, the prime differences between Chandrayaan 1 and Chandrayaan 2 and Chandrayaan 3 is what the objective of the mission was. When it comes to Chandrayaan 1, the mission was just an orbiter mission. Orbiter means it will go, move around the sun, find out what, sorry, not the sun, or move around the moon, find out what observations exactly are happening over there based on certain sensors and cameras, etc. Starting from Chandrayaan 2 onwards is where we've started having additional functionalities. So Chandrayaan 2 was a orbiter, lander and rover mission. Okay, so Chandrayaan 2 was an orbiter, lander, landing, and then there was roving, moving around on the surface. Now the important point to be noted over here was that when you have your orbiter, lander and your rover mission, here the lander mission and the rover mission failed. Which means even in Chandrayaan 2, what was active was your orbiter mission. Now, because this was the case, when it came to Chandrayaan 3, what did they send? They did not send a orbiter, because orbiter, the function we've already achieved success in. So, in the Chandrayaan 3, what you had was a lander and rover mission. This is important. These are the differences, prime differences between these parts. Once you understand the prime differences, then what exactly is the difference between 2 and 3? Let's have a look. When it came to Chandrayaan 2 and Chandrayaan 3, one of the first things that you will notice over here is the payload, the weight of the missions. When I have to compare, Chandrayaan 3 is a much more heavier mission than Chandrayaan 2. Okay. Again, the landing site was again here when it came to Chandrayaan 2, it was between two craters called as Mansimus C and Simplius N. Here, the landing site was not between craters. It was a much more larger landing site. This was actually done so that it can be error proof. So for example, Chandrayaan 2 had a landing site which was just 500 by 500 meter area. Chandrayaan 3 had a landing site which was 4 kilometer by 2.5 kilometer. 4 kilometer by 2.5 kilometer means the length and the breadth is what they are measuring. 4 by 2.5, which was the largest site which was there. Okay. Then coming specifically into some of the features of Chandrayaan 3. Chandrayaan 3, when it was launched, the scientist mentioned, or the chief officer of it mentioned, that Chandrayaan 3 is based, it's called as a failure based design. Failure based design doesn't mean that it's based on the failure or anything. It means that Chandrayaan 3 is designed in a way that whatever part of it fails, there will always be a backup for it in the mission itself. So for example, if the fuel fails, there's something else. So they have foreseen, or they've tried foreseeing what all failures could happen in the mission, and they've tried to curtail it based on that. Okay, this is also the reason why I want you to remember Chandrayaan 3 had much more fuel carried with it than Chandrayaan 2. Okay, Chandrayaan 3's landing velocity was much more higher than Chandrayaan 2. A new sensor was added called as your laser Doppler velocity meter. Then there was another sensor called as shape mission which was there. It has addition of more fuel. All of these things were done so that even if something fails, it will remain successful. So keep all these pointers in mind when it comes to Chandrayaan 3. Another important technology, now moving on to certain defense and etc. things which are there. Another important technology would be Kavach. Okay, so Kavach again was in news several times in the last 1 year. Kavach is a traffic collision avoidance system. As you can see, Kavach is basically something that is envisaged to prevent train collisions in India. And when you are saying train collisions, you have to not just you have to understand what the technology behind it is. This is based on your RFID tags, which is your radio frequency identification, which is there. So what happens is, trains which are running on similar tracks all would be equipped with RFID readers, and even the signals over here will be located with your. So this is based on radio frequency identification. So what happens is, whenever any trains which are supposed to be stopping at certain stations, or which have maybe the signal is faulty or anything, what happens is these RFID readers over here will read whichever train has overshoot it's if it's an exit, it has not stopped or whatever it is, and that will be communicated to a station TCAS, and using your Navic or your GPS or anything, they'll try to figure out where is the exact location of the train, and by then at the same time, using these satellites, whichever is the opposite train that's coming onto that track, they will be informed at least to not move further or to change tracks or whatever it is. So the point is, all of this is done via radio communication. So it helps in the two-way communication between the station master and the loco pilot. He is the driver of the train. This method was researched by Research Design and Standards Organization, RDSO, in cooperation with Indian industry. Okay, so this is to prevent train collisions. The important thing over here is something called as SIL. SIL primarily is a safety integrity level, and safety integrity level primarily is a standard that's given by owners, operators, etc. Now, this is not a government, category or anything. But generally in industries, to identify if a certain technology is really error-free or not, you can use and see if what SIL tag it has. SIL 1, 2, 3, 4. 4 being the highest. Now, Kavach system has been given an SIL 4 tag, which means that it will have a higher protection given. It will have a higher, or it will have a much lesser error. So you can see over here, Kavach is one of the cheapest SIL 4 certified technologies where the probability of error is 1 in 10,000 years. So that is the lack of error or the error proofedness of your Kavach system. So again, that is important for this year. Another important defense related finding of this year is your Agni Prime. Now, Agni missiles are ballistic missiles which are surface to surface in feature. Now, what has happened over here is, over the time we've had Agni 1, 2, 3, 4, 5, 6. Okay. Now, Agni 1, 2 and all of these missiles are generally used for shorter ranges. Okay, though it's called as medium range ballistic missiles, the range which is maybe Agni 2 up to 2000 kilometer, you can have it. Agni 1 is 1000 kilometer, 900 kilometers, etc. So now we've created something called as Agni Prime. Agni Prime is actually a missile which has the maximum range of 2000, which means that when Agni Prime comes into fleet, it will replace the Agni 1 missile. Agni Prime is also important because it's nuclear capable. Next important thing is, leaving aside all of these features, Agni Prime has something called as a canister launch. Canister launch means the missile is created in such a way where already whatever is the warhead or bomb or whatever you have to carry will come fused within its structure. That means you don't have to spend time when you have to attack some place immediately. You cannot be like, okay, fine, let me go get the bomb from the storage unit. In the missile itself, this is placed and fused into its structure. That's why it's called as a canister launch, and you can swiftly transport it to other areas if needed. And you can even launch it in a similar way. Okay, so this is the feature of Agni Prime. Moving on to one of the most important last areas of this discussion, communication and IT. I would say in communication and IT, one of the most prominent current affairs that you have to be aware for this year is ONDC. And a lot of people have confusion as to what is happening in ONDC. Is ONDC an app? No. Is ONDC any other kind of service provider? No, that's not what ONDC is. So, to understand ONDC, one of the first things that you have to be aware of is something called as open network. Okay, open network. What exactly is an open network? So, understand, whenever we have to create any kind of system, so generally this is how our, like if for example, there is a seller, Amazon. What Amazon will do? Amazon will create its own app, which is the Amazon app, which is there. So, the sellers will have an Amazon app. The buyer, which is me, I will also have an Amazon app in my phone. And if both of us have the Amazon app, the app has to be created. That is an important factor over here. It's not a simple process. So the app has to be created. The buyer gets in touch with the Amazon app. The seller is also in touch with the Amazon app, which means if a product has to be sold via the Amazon app, both the buyer and the seller have to have the app. And then they can buy it. Okay, so you do realize there's a problem with it. Not everybody has if you have someone who has some folk art or something and they want to sell their products, and Amazon, when you are a seller on Amazon, they will take, because they've created the app. The app is what is bringing all the people together. So they'll be like, see, you're putting your stuff on my app, so you have to give me some money for it. Not all sellers can do it. Okay, so here is where an alternate idea comes about, where instead of 1 or 2 apps, you create an open network. An open network is like a plug and play model. Plug and play model means if I am a person, I have a good product that I can sell on the internet, through the internet services. But I don't have the money to create an app. I don't have the technological expertise to create an app. So what ONDC does is, ONDC will create all the necessary setups. So ONDC will first ask you, are you selling food or electronics or whatever? So I'm saying I'm selling electronics. Tick. Then ONDC will be like, okay, for electronics, this is the kind of service that we have. And instead of creating an app, ONDC will list you amongst these electronic selling different providers which are there. So I don't have to create an app. I can sell my products without even creating an app. This is actually a free, it's an open network, which means I don't have to pay ONDC anything. I can just list my products. Consumers can come over here. Consumers can see my electronics app. Electronics app, when I'm saying my electronics products, several other electronics products of different sellers also, they can choose, they can buy. It has become, or ONDC would ideally democratize the e-commerce space. Earlier, only if I had the money and the technological expertise could I create an app, or if I had the money, I could get onto all the existing apps. ONDC has just let it open. Like, you have a product to sell, come, we have a network that you can just plug your thing into and sell it. So ONDC is the newest addition into India's digital public infrastructure, which is an expected question for this year, primarily because in G20, India has now taken the initiative to list all our digital public infrastructure so that other G20 countries can also use it. So this is the reason why you see not only G20 countries, other countries all in general can use it. Singapore is now having UPI payments. You have certain countries in Africa that are now taking up Aadhaar related platforms. How are they all able to do it? Why? Because we've created digital public infrastructure. Infrastructure generally means what to you? Road, railway and all of that. Digital infrastructure means what? Something that's created on the internet. Digital public infrastructure means what? It's open to whoever wants to use it. So digital public infrastructure in India can be created by completely government or private, or government and private together. In this manner, existingly, we have Aadhaar, Digital India Mission, PM Vani initiative, unified UPI which is there, CoWIN platform, your network to villages, Bhashini platform, India Stacks, Sanchar Sati, all of these are digital public infrastructure which is open to all. Now, I don't mean it's an app. This is not, none of these are apps. Tomorrow, now look at this, okay? India Stacks is an online global public digital goods depository to ensure no one is left behind, which means anyone who, there's any agriculture product, any medicine, whoever wants to list their products and use this. Now, it's named India Stacks. Tomorrow, if Bangladesh wants to use it, they can use the India Stacks platform and they can ensure that all of the goods in their country have a correct repository. Like, okay, in this much of markets, this much of vegetable is available, this much of, you can use it across the globe. Understand? So this is an important area. They could ask you maybe a very simple question, like they'll list 3-4 things and ask you which is part of India's digital public infrastructure. I've given you the list. ONDC is also part of India's digital public infrastructure. Okay. Another important mission or other important technology, by, as you can observe over here, I am talking about the technology that is indigenous, and indigenous technology in itself has a question scope. Okay. The next thing that we have to discuss over here is D2M technology, which is your direct to mobile technology. Now, the idea of direct to mobile technology is in a way that, so now on your mobile, you get access to products or you get access to services when you have internet connection. But what if you don't have internet connection? Okay. Or what if you are in a remote area, but the government wants to disseminate some good quality content to you, something informational, educational, etc. So, direct to mobile technology is a method by which videos and multimedia content can be directly portrayed on your mobile phones without active internet connection. For example, it's like a radio but for pictures, because in a radio what happens, you tune into a right frequency, you get the content. You don't require internet for a radio to work, I mean an actual radio. So at the same time, imagine you are tuning into a certain frequency and you get video content. And what is the frequency you have to tune into? 526 to 582 megahertz. When you tune into that, now please don't try it, it is still underway, okay? So when you tune into that, what is going to happen? You can have content, video content that is made available to you without internet. So it can be any entertainment, educational, informational, etc. The idea of this is any internet spectrum which is there, it can be freed because a lot of content can be provided in this manner. Okay, this is undertaken by your Prasar Bharati, and that is why this is important also. It's the Press Bureau of India, so it's undertaken by Prasar Bharati. One of the last important topics that we have to discuss right now is something called as Web 3.0. See, I am telling you the PDF that is going to be shared along with this class and video has a lot more content that you can just go through, but anything that requires explanation and that requires clarity is what is being dealt in this session, so that, you know, those who are very new also can benefit from it. Those who are interested can just go through the slides. The slides are pretty detailed also, and it will not take much of your time if interested. It's not a necessity at all. Okay, the most important topics are what we've discussed today. Okay, now coming to a topic of Web 3.0, you will understand over here that currently Web 3.0 is all the buzz. There's a lot of noise about what is this Web 3.0. So all of the things that we are seeing right now, your chat GPT, your blockchain technology, all of these innovations are part of something called as Web 3.0. So then what is this 3.0 and what was the previous versions of it? Web 1.0 and Web 2.0. Web 1.0 was something that was a read-only mission. What was read-only meaning? Read-only means you could not communicate anything to it. Whatever was provided to any platform, you could not even ask for it. So there would be already some content you can read only. The internet had just static web pages. That was Web 1.0. It was not continuously changed. Web 2.0 is a read and write internet. Read and write means we were also able to communicate with that, which means that if something had to be changed, if we wanted some specific content, we could ask for that, and that would be what we would receive. So Web 1.0 and 2.0 just opened the idea of communicating with the internet. Web 3.0 is something called as read, write, execute. Read, write, execute means, see, now, until now, what we were using was there would be a server. The server would give us some content. We ask the server for something, the server will provide us. This was Web 2.2. Now, the idea is, let's not even have servers. Anybody who wants to give the content, let them give the content. When I'm saying content, I don't mean your blogs and YouTube and everything. The idea is, where are you storing your content? Where are you retrieving it from? So Web 3.0 is a system of decentralization. Decentralization means that you will be, and some of the features of it is, see, for example, you're having blockchain, you're having edge computing. What is all this? You are not relying on one single server. You are relying on anonymity. Anonymity is another feature of it. So machine learning is another feature. Permissionless, meaning that each individual can access without governing body's permission. Also, trustless. Don't get this confused. Trustless means participants who are there on the system are going without any trusted intermediary. That means if you're going on a blockchain, which you are aware of, what happens over here? You're getting onto the internet and you are in the blockchain technology. There is no RBI or anyone governing over it. There is no intermediary that says, okay, this is valid or not. So Web 3.0 is all about decentralization, trustless, permissionless, machine learning, and connectivity. The further feature of this is Web 5.0, where it will involve even higher order things like your generational AI, etc. Generational AI will be part of 5.0, not 3.0. Okay, so I hope with this you have some clarity on the important topics that you will have to look through when it comes to UPSC for this year. And again, understand, UPSC is not going to exhaust you with very weird science and tech current affairs. They always are bothered about the crux, the basics of a lot of these things. So whenever you hear a technology, if it's a satellite thing, look at what is the frequency of it. What radio are they using? Radio frequency, microwave, what kind of things? Can they reach out to people? Can it be blocked? All of these basic features is what they look. And again, it's very close to the exam. So don't study anything new. If something is there, as what a mantra that I used to always keep in mind is, and you have to have that when you do UPSCs, and obviously towards the closer times of the exam, you will have several friends of yours asking you, have you done that? Have you looked at this technology? Have you learned that thing? My point at that place would be, okay, if I have not looked at it, it's not going to come for the exam. Okay, because trust me, it might not be an actual good philosophy or anything, but 7 days into the exam or 6 days into the exam, it's better that you do not burden yourself with such newer thoughts. Understand? So if you are hearing something absolutely new, just be like, okay, if I have not heard it, it's probably not worth looking at it right now. Okay? So there is, the point is, if it's something very complex, if it's a small fact, I'll obviously look, like the first moon mission or something like that. It's a fact means fine. But if it's completely new theory or some new textbook or some new material that is out there and people are like, have you read that? Please don't do it. Please don't do it at this point. Okay. So I hope you all will do the exam very well. Let me know the comments in the comment section, which is I keep reading all your comments. This is the reason why I'm able to respond to some of the comments that have been put over there. Okay, so I hope you do the exam very well. Take care, happy learning, and all the best.